our fourth webinar of our webinar series in this summer season. And we're very happy to welcome Dr. Sharmeen Roy, who's joining us this morning from the US. So it's morning for her, afternoon for us, and we're very happy to have her here. Talk a little bit about precision dosing. So she's going to talk about bringing complex modeling to the bedside, improving patient outcomes with model-informed precision dosing. Dr. Sharmeen Roy's 20-year career spans pediatric clinical pharmacy, clinical research, pharmacogenomics, and strategic leadership. She received her doctorate, a Doctor of Pharmacy degree from the University of Illinois at Chicago, and completed her pediatric specialty pharmacotherapy residency at Texas Children's Hospital. She is board certified in pharmacotherapy and holds a certification in pharmacogenomics from University of Florida. Her tenure includes esteemed institutions such as Texas Children's Hospital, University of Chicago Medical Center, Nick Laus Center Hospital, and Miami Cancer Institute. At PipelineRx, she held senior roles managing large teams and driving client engagement for over 500 hospitals. She is actively involved in CPIC, Stripe, PIDS, ACCP, ASHP, and PPA, and being a practitioner who embraces technology, Sharmeen strategically blends extensive clinical expertise with cutting-edge innovations, leading the way in patient care. I would like to welcome Sharmeen. Thank you very much for bringing your expertise to the floor today, and we very much look forward to your presentation. I'll hand the floor over to you.
Thank you, Madre. It's a pleasure to be here. Let me just make sure my technology is functioning correctly here. Let me share my screen. All right. Hopefully that you can see my slides.
All good. We can see everything.
All right. Perfect. Well, thank you, Madre, for inviting me to speak to the group here. Today, I'm really, really excited to be talking about this topic. I'm very passionate about precision dosing and technology and combining the two in clinical care. So good morning or afternoon, I should say, everyone. It's morning for me. We are living in a very exciting time right now where precision medicine is no longer just a concept, but it's a practical approach that can significantly improve patient outcomes. At DoseMe, we have developed an advanced software called DoseMeRx that leverages complex pharmacokinetics and pharmacodynamic models to provide precision dosing and recommendations. And it's done right at the bedside. So clinicians can utilize it in real time and see the impact in real time as well. So our goal is to really enhance efficacy and safety of treatment by tailoring those doses for individual characteristics, as well as ensuring that each patient receives optimal therapeutic benefit. So to begin, I really want to discuss the need for precision dosing. In modern healthcare, traditional dosing methods often rely on population averages, which do not account for individual variability and in-drug response. So we know from all the evidence that is out there that there's a lot of variability between patients. I may metabolize a medication very differently than you all may. So we need to make sure that we're getting to the right dose for the individual rather than looking at a population average. And because of this, the one-size-fits-all approach really can lead to suboptimal outcomes, which includes adverse drug reactions and therapeutic failure. So precision dosing is, I don't have to convince the group here, but precision dosing really aims to tailor treatment based on individual patients. These are my disclosures. So just to give you an overview, we'll be talking about MIPD or model-informed precision dosing, how we're bridging the gap or closing the gap, I should say. I'll mention some, success stories that we've had, looking at some special patient populations, as well as talking about advances in beta-lactam dosing. And ultimately, how do we overcome some of these challenges that we have and barriers we have to implementation, and what's really next on the horizon. And, like I mentioned earlier, that I recognize there's probably no need for me to convince the audience here on the importance of, integrating technology into clinical care. However, we have to acknowledge that despite the advances, it continues to be an uphill battle. So I always like to start my presentation with a question you can think about as we talk through the various topics today. So how can modern advances in technology and software impact clinical dosing decisions? And what steps must we take to fully embrace this inevitable transformation? You can see from the slides, you're probably wondering why I have these images on the slide. So we want to really acknowledge how we've integrated technology. And when we talk about technology in today, we could automatically think about software and AI and all sorts of more complex technologies. But let's just take a step back because technology is something that has been part of healthcare and part of the innovation in healthcare. So in the images you'll see there's on the top right here, or sorry, the top left here, where a physician, before we had the invention of a stethoscope, vitals were uh, by a physician was checking vitals by just listening to the patient, putting their ear to their chest and counting. And if you imagine doing that in the current day and age, that would be very difficult. And we can't really imagine a world where the stethoscope wasn't available. So technology isn't just software, it's a lot of different inventions we've had in healthcare. And on the right-hand side, you'll see what a thermometer, the evolution of the thermometer, and how taking a patient's temperature has evolved. We don't, as we know from recent years, that we don't even need to touch the patient anymore. All you do is point to the forehead, and you'll get a temperature reading. So I really quickly want to go over some terms you'll hear throughout the talk, just so everybody's familiar with them. I'll refer to models, which are pharmacokinetic models developed and validated from population PK studies, in which the study is defined by mathematical equations that describe the time course of drug exposure in that patient population. Covariates, which is a variable or patient characteristic that's identified in a PK model or a study as a source of interpatient PK variability in patient exposure. Bayesian method, which is an approach to incorporate PK model in simultaneously integrating patient covariates. And this is the method, the dosing method we apply in our software. An MIPD model-based approach, which is also referred to as model-informed precision dosing. So what exactly is model-informed precision dosing, or MIPD? It's really not a new approach. In fact, it's been something that was first developed in the late 60s. The FDA, the EMA have required population PK studies from 1990s onward. So, these PK models are part of the drug development process. Any new drug application, approvals, clinical trial clearance, and design incorporate PK design and PK models in order for a drug to get approved. And now we're seeing that the wealth of knowledge that has been developed in the drug development process has been entering common clinical practice now. And on the right-hand side here, we have just a timeline so you can see how it's evolved and how it's been incorporated and where we're headed. And I did include a picture of Thomas Bayes. He is the mathematician who, developed the Bayes theorem, which was a formula that is determined by conditional probability. And we'll talk a little bit about Bayesian dosing methods. So I just really wanted to include a little bit about him here. As he's kind of the foundation of the theorem of the Bayesian dosing method that we apply in our software. So how do we bridge the gap? I think we're at that point, we're closing the gap. And we just have to, again, remember that we take an oath as in healthcare to first do no harm, which is a core fundamental in patient care. But in clinical practice, we're still seeing therapeutic failure and toxicities, which we want to try to minimize or avoid altogether. And because model-informed precision dosing does involve application of mathematical and statistical algorithms, it's often perceived as a complicated and time-consuming task. So what we want to do is close that gap and use, our software, technology, clinical advances to enable that translation from research to bedside, and really provide the tools that clinicians need to apply it to patients' bedside. And as we know, there is no shortage of evidence in the literature that's using the technology to get closer to precision dosing, and individualized dosing improves outcomes. There's plenty of studies. This is just a few that I highlight, but if you do just a quick search in PubMed in precision dosing or individualized dosing, you'll get many more papers that have established and supported that precision dosing and individualizing dosing is really what we need to get to for all our patients. So how does it work in our in the software that we have, DoseMeRx? It's combining, the inputs are the patient characteristics, and it's combining that with the clinically validated PK/PD models, and providing those individualized dosing recommendations. And that is very important that it's happening in real time, because you're taking in, we have a lot of data that comes in through our lab systems, our electronic health records, and constantly, updating with new results. And we want to incorporate that as we're updating and adjusting the patient's doses. While that was already being done by clinicians at the bedside, using, more manual applications, but the software really does make that more efficient and streamlines the process, and which allows and frees up the clinicians to spend more time taking care of the patient and spending time with the patient. And this is just an illustration here of what the dosing curves, and I use vancomycin as an example, and we'll cover that in a second here. But here you'll see there's two dosing curves, and since we implement a population PK model, which then combines patient data, you'll see the red line is what the population PK is, and as you get more patient information here, the blue line is the individualized curve, and the X's here are the actual concentration and the serum levels. So the reason I am starting out with vancomycin is because vancomycin is one of the single most used antibiotics in a hospital setting. And it's also very well known for its adverse effects, particularly nephrotoxicity or acute kidney injury that it causes. And because of this, in 2020, there were updated consensus guidelines which were published with several different organizations came together and published an update to the therapeutic monitoring of vancomycin for serious methicillin-resistant Staph aureus infection, or MRSA. And the key recommendations from those guidelines were that in adult and pediatric patients with suspected or definitive serious MRSA infection, an individualized target of AUC-MIC ratio of 400 to 600. The monitoring recommendation was to begin with the first 24 to 48 hours. And the traditional method of using trough-only monitoring with a target of 15 to 20 was no longer recommended, because based on efficacy and nephrotoxicity data in patients with serious infections caused by MRSA, that was no longer recommended. They also included some key monitoring recommendations, and in which the preferred approach that was recommended to monitor AUC through the use of Bayesian software with embedded PK model based on richly sampled vancomycin data as the Bayesian prior, using 1 or 2 vancomycin concentration with at least 1 trough. And this is very important because this really kind of was served as a catalyst for clinicians to look at software and incorporating the software into dosing decisions. There were studies that showed that trough alone can generate accurate and reliable estimates for daily AUC. And I forgot to mention the AUC that I'm referring to is area under the curve, which is a time concentration curve. However, no guideline is complete without providing alternatives. So the alternative approach was first order kinetic calculation, which are done more in a manual basis. So with these recommendations, that really shifted how the recommendation was for monitoring. And what we wanted to know, if you notice in the guidelines here, these are in 2020. So 2020 was, as you all may remember, a busy time, and we had different priorities at that time. It was during the pandemic, and everybody was focused on that as a priority. So it shifted. So what we decided to do is see what really people are doing, and we thought we'd look at, in last year, if you notice, in 2020, because of the pandemic, things were kind of put on hold, and really, we didn't see a lot of movement as far as implementation of those recommendations. So we thought that we would see what is actually happening, and Dr. Mark Sheets, who is an expert in the field, conducted a survey. And, we all know where the most effective surveys are conducted. This was done on a very reliable tool known as X, or formerly known as Twitter. And here are the findings. So as you can see, we wanted to know, what are people doing? How he asked the question, how are you dosing vancomycin? And a large percentage, 50%, over 50%, were still using the conventional trough method. There were some, about 11%, that was using AUC population estimates, about 16% doing 2-level, and then about 20% using a software or using Bayesian methods. So because of this, as you can see, the guidelines were still not fully implemented. I think trough-based dosing was something that has been ingrained, as a pharmacist who trained using the trough-based method. It is very difficult to change that, and many people don't have the tools to be able to monitor based on AUC. So what we did was, we thought we should really see what's happening. So we wanted to look at real-world sample of the data for patients that were dosed using Bayesian software. And this was data that was presented at the most recent ECCMID meeting in April here of this year in Barcelona. Dr. Sheets was, kind enough to let me borrow some of his slides that he presented at our abstract. So we presented big data Bayesian truths. No vancomycin trough concentration target is sufficiently precise for safety or efficacy. And really the purpose of this was to look at a large cohort of internationally represented real-world patient data using Bayesian estimated vancomycin AUC exposure versus measured trough concentration, to gain a little bit more confidence for our users that, really that change from trough versus AUC, it should be done. You cannot use that as a trough as a surrogate. And we assess the categorical agreement between those two groups. This was including all adult patient populations that were treated with vancomycin in 2022, and with therapeutic drug performed as part of their routine clinical care. We did exclude patients that did not have any plasma concentrations or were on hemodialysis, and those that had less than 1 day of treatment. There was data obtained from unique patients, and dosing events were quantified. We looked at trough concentrations, or Cmin, which were identified, and patients' individualized AUC was calculated using the Bayesian method with 4 validated models. A primary analysis here shows the characteristics. As you can see, this was a large number of subjects. There was over 35,000 patients here. We did, and when we initially looked back 2 years, it was actually even much larger. So we decided to look at 2022, and it was over 35,000 patients and over 283,000 courses that we analyzed. And here was our finding. So we looked at assessed the categorical agreement between the two. So we looked at concordance between AUC and Seaman status. So in this graph here and in the categories here, the red ones are the most worrisome, but fortunately it is low incidence. But I do want to highlight here the low trough but goal AUC patients here. And you'll see it's about 30%. And what that means from a clinician's point of view is that based on a low trough, you're likely to increase the dose inappropriately. So when you receive a low trough, you are trying to get them to therapeutic level, but they may already be at target AUC, so that increase in dose may not be warranted. But when a trough-based method is being utilized, you may inappropriately increase the dose, which can then lead to adverse effects, specifically looking at nephrotoxicity. So the summary of our findings was that only 23% of the paired Seaman and AUC were at target, which means there's only 23% of patients where you could use the trough as a surrogate for AUC, which demonstrates again the variability of exposure in this patient population. There was categorical disagreement between Seaman and AUC, which was about 33%. So a third of the patients there was disagreement, and the most disagreement being for those 29% where the low trough, the patients had appeared to have a low trough, but they were at target. And this is the one where clinicians were at risk for overdosing patients on troughs alone. Now, 30% is, if you think about vancomycin being one of the most commonly used antibiotic, 30% can mean a lot of patients, and that can mean a lot of patients that can potentially have adverse effects. So these findings really ultimately support the recommendations in the guideline, supporting that AUC therapeutic drug monitoring and challenging the trough-based methods for adequate vancomycin AUC proxy. There have been other recent publications to show improved outcomes with real-world applications of our software. So on the top here is the Covington study. Dr. Covington looked at acute kidney injury incidence using either the two-level method versus the Bayesian dosing software. And the findings from the study was that there was definitely a significant decrease in acute kidney injury, an increase in target attainment. So patients were able to get to target therapy quicker. And that's very important for antibiotics because you're trying to make sure that you can, get the infection under control. And you can only do that with our adequate target attainment. And the third finding was there was an increase in usable levels. I believe it was about 96% of the levels were usable. And that's important because oftentimes when you're monitoring for vancomycin, if the trough is not measured appropriately, you oftentimes have to repeat it. What that results is it delays patient care. It increases the number of assays you have to do and ultimately increasing the burden on nursing staff as well as laboratory personnel. The second study here is at a cancer center in Brazil. Oncology patients are very unique. Their pharmacokinetic varies quite a bit. And it is really, really important to get the dosing right in those patients. And in Dr. João Teles, in his study, he found that there was a 70% decrease in the rate of kidney injury, which is significant, which had a huge impact in the cost savings that were realized by that cancer center. And one of the other bigger challenge is special populations. So, the patients that we are treating are changing. They're becoming more and more complex. There's not a lot of dosing information that is available for those special patient population, and that's what these two studies highlight. So on the top here is looking at a single center experience. This was again recently presented at ECCMID using Bayesian AUC-based dosing of vancomycin in pediatric patients, which showed overall decrease in risk of toxicity. And then second one, again, by Dr. Covington and group, looking at obese patient population and seeing if one level concentration combined with Bayesian modeling can adequately get to target attainment. And there was overall high agreement between 1 versus 2 concentration for AUC monitoring. So as you can see, not only is this helping individualized dosing for all, our general patients, but providing that more specific method to take care of the patients that are more, specialized, may have various comorbidities, as well as have, complexities that are not necessarily accounted for when a drug is going through their development process. And to further illustrate the impact of MIPD, let's really delve into beta-lactam dosing. So beta-lactams such as cefepime or meropenem, those are reserved for critically ill patients and are dependent on maintaining adequate concentrations. They also have a high variability, not only because of the pharmacokinetics of the medication, but also the patient population that often is on these medications, which is critically ill patients, which, as you can imagine, have a lot of changes. Their clinical status is changing quite a bit, and they may have fluctuation in their in their renal function, in their fluid status. So it really impacts how it impacts drug concentrations, and maintaining those are very, very important. Therapeutic drug monitoring is still not a standard of care. However, there have been some guidelines that have been published to recommend, in certain patient population where that can be helpful. But software can still help you take care of these complex cases. So whether you are doing, TDM or not, which is therapeutic drug monitoring, it does allow you to use targets such as % of time above MIC, which is the minimum inhibitory concentration, and that can help you guide your dosing for the patients. So as you can see here, this is just an illustration of what, how the software can help you do model-informed precision dosing with or without therapeutic drug monitoring. It'll help you manage some of those complex patients, and you can still utilize some of the patient's data to individualize dosing with, even if you don't have the the drug concentrations obtained. There are other applications as well. So we've seen significant advancements in infectious disease, but we have learned from it, and the benefits of model-informed precision dosing extend far beyond the antibiotics. There are 2 drugs here I'll highlight, the first one being busulfan, which is used primarily in conditioning regimens for bone marrow transplantation. We looked at there was a retrospective review at a large hospital hospital system in Texas. He looked at about 24 patients who were receiving bone marrow transplant. And busulfan is a medication that requires therapeutic drug monitoring. You have to really closely monitor exposure and get to target cumulative AUC, which requires, some centers will do from anywhere from 8 to 10 10 levels on a patient for every dose, which you can imagine is a lot of blood collection, blood sampling, and a lot of time that is involved in it. Using the software can really minimize that and simplify it in a safe manner. We've seen centers that have been able to reduce it to 2 or 3 levels. And as you know, in pediatric patients, they've been able to reduce it to 4 or 5 levels, which is significant. So reduced requirement for blood collection was found as a result of this review, which also lessens the distress to the stem cell patients who are getting conditioning regimen for an upcoming transplant. And ultimately, it also, again, decreases the burden on not only staff, but unnecessary lab assays you have to do and decreasing the cost. And the second drug I'll mention here is infliximab. Infliximab is really the cornerstone of the treatment of immune-mediated inflammatory diseases. And therapeutic drug monitoring is becoming more and more prevalent in this drug, as well as other agents that are used in this, in this patient population. We can use this therapeutic drug monitoring and individualized dosing. It's been recommended in some of the guidelines. There has been evidence that it's more cost effective than doing empiric dose escalation based on response. And improve clinical scores, including, inflammatory markers and overall outcomes during the induction phase. And infliximab is one of those medications that, you can develop antibodies to it, which can reduce its efficacy, and it can also impact the concentration of that medication. So what using a model-informed precision dosing does is that it helps you prolong the use of that drug and lessens the discontinuation, which is oftentimes, when a patient has to discontinue, they have to switch over to another agent, and it can become a trial and an error process. So this is really informing those dosing decisions and adequately adjusting it so we can get not only prolonged use of the medication, but get patients to more of a maintenance phase and remission. Now, despite its benefits, implementing model-informed precision dosing at the bedside does come with its challenging. There's, logistical, educational, and technological barriers. There are strategies that can be used to overcome these barriers, and that first and foremost involves just an interdisciplinary collaboration. We are no longer able to function as a single clinician. We work in teams, take care of patients in team, and we really have to collaborate in that manner. And when you're, thinking of implementing this into your clinical workflow, you really need to engage all the stakeholders, really get your support and identify your key champions that are going to help you make the case. And really need to highlight the added benefit for interdisciplinary teams. How this decreases having this implemented into the current clinical workflow will, decrease the burden on teams. We're all expected to do more with less resources. Post-pandemic, it's really put a stress over health systems due to the shortages that we're experiencing. So we should use these tools to really alleviate some of those stressors and utilize the data that we can obtain from use of technology and metrics that we can have pre and post implementation to really show the value in utilizing this and expanding the use of software like this. We have to continuously educate and train all staff. It's not just the pharmacists that have to be the champions. We have to get nurses, physicians, and other healthcare providers all on board in this approach. And you have to, with any program that you implement, it's really important to continuously monitor and make sure you're updating and revising your protocols based on what your findings are, really showing the value by demonstrating safety metrics, with the example I'll use of vancomycin is showing the decrease in your AKI rate, decreasing the patients being outside of the range of therapeutic targets, which can then reduce the number of patients that are experiencing toxicity and overall show the improvement in outcome and outcomes data. So what's next for model-informed precision dosing? We have to, again, we need to continue to ask the question, what is the right dose for at-risk population? Whether it's the critically ill population, the elderly, obese, pediatric, oncology patients. There's a lack of labeling and guidelines in those patient population, and that's where in my opinion, it makes the most impact in those patient population because you're not going to be able to go to the label or the package insert to find recommendation. The FDA has also, has launched a Project Optimus, which really shifted the paradigm for dose escalation in the oncology drug development. So, really asking pharmaceutical industry to look at not the one-size-fits-all approach, and not the maximally tolerated dose approach, but really looking at other, using model-informed simulations to recommend the most appropriate doses, which will help because this will focus our dose based on exposure rather than looking at the, one-size-fits-all. And we will be able to utilize, this shift in paradigm to confidently transition the developed models that are developed during the drug development process and apply to real world. And we need the software to help make that translation. The other area that is very important to dosing is pharmacogenomics. It can have a significant impact not only on the drug selection, but also pharmacokinetics. So how you are metabolizing a medication, how you're processing the medication, your pharmacokinetics, your pharmacodynamics, and really integrating pharmacogenomics into model-informed algorithms, model-informed precision dosing algorithms will help us further refine and personalize dosing regimen. This is something that's, again, not new. It's already being incorporated in some population PK models that have been established. I've listed some of the medications here, tacrolimus and CYP3A5. That tacrolimus is a drug that is used for solid organ transplant patient and some stem cell transplant patients. Voriconazole, which is an antifungal. This one has some therapeutic drug monitoring that is happening, but combining that with pharmacogenomic results can really help tailor that dosing. It's an antifungal that's used in not only critically ill patients, but also immunocompromised patients. And lastly, warfarin. Warfarin has had pharmacogenomics that has been established for many years now. It's an anticoagulant. It's a drug that's been around for a long time and it's not going anywhere. It's still currently used widely, and has been associated with the dosing, and the dosing ranges have been associated with the Pharmacogenomic Association. And again, I always go back to this. Nothing will expand, and adoption will only increase with education. So we need to continue to educate our colleagues and other healthcare providers and continue to increase awareness. And with that, I thank you for your attention, and I'll turn it over to Madre to see if there's any questions.
Thank you very much, Sharmeen. Was very interesting, as we well expected. And we haven't got any questions yet from the audience. But I would like to know, can you tell me which other drugs can we use this precision dosings for?
Yeah, so, models are something that we're continuously evaluating, and we really want to, add models that are available are currently being, have narrow therapeutic index. So we currently have about 46. The number keeps increasing, but there are drugs in solid organ transplant and oncology, in other anti-infective agents like aminoglycosides. There's newer guidelines that are available for all these agents to do more AUC guided dosing, which is requires the use of software. So and that really the space that I see more and more need for this is in the oncology drugs. I think we've all kind of realized and accepted that the pharmacists were kind of champions of this, but that really tailoring the dosing to patients and their whether it's their organ function, renal function, is very, very important for that patient population, and it's a very um, vulnerable population, correct? So it's, there's not a lot of data. We want to minimize toxicity. In the oncology patient population, they're experiencing toxicities not only of their disease, but the drugs have a really high risk. So those are some of the areas that where we have existing models for, and we continue to expand on.
Thank you. Thank you very much. We have a question from Zaina. I have opened her mic. If she wants to ask you directly, she can just switch her mic on. We'll give her a second to do that. Otherwise, I'll ask the question. Oh, there we go. Hi, Zaina.
Hi.
Welcome.
Thank you. Hi, Sharmeen. Thank you
Hi.
for the informative talk. It was really interesting. And I just wanted to ask first the question that Madri asked you about before, but I'll ask my second question. Is the ethnicity or the ethnic differences include as a factor, a variable in the models that you build? Are you considering the variability in this variable in your models?
Yeah, that's a great question. So so when we go through the selection of the model, we acknowledging that we do try to select the model that has the widest cohort of patients. Now, with that said, in an ideal world, it would be a cohort that includes patients from all parts of the world, and that's just not reality. So when we do evaluate that, we do go through an extensive internal and external validation to see its applicability in different patient population. And many of these models are already, even if they've not been developed using, a certain cohort from a certain part of the world, over time it does get utilized with our, international sites. And the thing to remember is that PK variability is captured when the dose is individualized. So if you recall the curves that are shown in the dosing profile, there's a population PK, which is the model that's implemented, but it's always taking into the consideration the patient's data. So whether it's characteristics, their renal function, their blood concentrations, it's incorporating that and individualizing the curve. So it does correct that and individualize that over time. Now, again, with pharmacogenomics incorporation, I think that will become more of a focus to ensure that it's being incorporated or accounted for. And that's in the works. So, more to come on that.
Thank you. Thank you for the very clear answer. And for all the presentation, it was very impressive. Thank you.
Thank you, Zaina.
Thank you, Zaina. Also, so Sharmeen, I know that one of the questions we wanted to discuss is if the software can, and how it can integrate with electronic health records. I know that this is quite new in the UAE. We have electronic records. This will be implemented as it goes further. I know in Saudi, they're also implementing it strongly. Is this a possibility?
Yeah,
to integrate
so we
this,
do.
or would it
Yes.
be freestanding?
Yeah, so and I've been a pharmacist, and I've been one of those pharmacists that I don't want to look at 5 different platforms to take care of a patient. So we want to make sure that if you don't make it easy for clinicians to be able to access it which is, if they're working on an electronic health record, you have to make this available there. Otherwise, it becomes challenging and becomes another barrier. So it does integrate. So we have a version that is available via the web where it's not integrated, it's freestanding, and you can, that requires manual entry of data. But it can integrate with, Cerner, Epic. We have some surveillance systems that it also integrates with. And what that does is make it again, streamlines the process where it automatically populates and pulls in the patient information. So you don't have to do that manual entry. It decreases the chances of error in that, data transcription. So it does integrate with some of the major EHRs. I know that I believe in the Middle East, Cerner seems to be, I think Cerner and Epic kind of are the main ones that are being explored. There are others, but we are, the way our technology is developed, it is able to interface with any system that has, APIs built in. So it's easily doable with other systems as well.
Thank you. Zaina has another question. Zaina?
Yeah, thank you for this chance. So I understand that you have mentioned to me in my previous question that the patient's data will be integrated, but building on the algorithm in one like homogeneous cohort will make it biased towards this cohort. That's what I meant. Are you considering like building your algorithms on more heterogeneous cohorts coming from different countries, different populations, and all the variables are considered in the first cohort that is built based on it, the algorithm is built. I'm not sure if I'm clear.
Yeah, I think, no, it's clear. I think I know what you're asking. So we have to, due to some regulatory requirements, we have to go through an extensive process for the validation of models. However, what we are able to do, and if a hospital is interested that, so this is a population model, and now we have data, can we modify this, or can we come up with a a model that is more representative of the patient population. So we can, once you start utilizing it, once we have enough of the data for that site, we can work with the site to, modify it to your patient population. So identifying if it's, re-estimating it for your patient population. And there's, different methods depending on the model, how that can be done, but that is something we can certainly offer, that if that is an interest of the hospital, that we do consider that. It just requires having enough data to be able to do that exercise.
Yeah, so I got it. It is a flexible model, like it can be.
Yeah. Yep.
Great. Thank you.
Mm-hmm.
Sharmeen, I just also wanted to ask you, you just said when we were just discussing before Zaina's question, that you can apply this anywhere where there's EPI already built in. Does this mean that is to have that in place is a requirement? Because I know from having the regulatory events, I know EPI is only starting now in, say, Egypt and other countries.
Oh, so I meant Fire API. So that's a way to integrate. Yeah. So that is what.
EPI. Okay.
Oh, sorry about that. Yeah. No. So
no, my mistake.
The way it integrates with electronic health records, you have to have a Fire API. So as long as that's a capability, that it could be done. From a regulatory perspective, we have the highest level of certification here in the US. It's, HITRUST certified. We are CE mark as well as TGA certified. So it's, those are just requirements that are very, very important. And, we comply with all of those, because I know those are remembering the patient at the core, patient care and taking care of the patient. Their data is also something that is very should be handled very carefully. So we, make sure that we are following the highest level of security with the patient data as well.
Thank you. Yes. So how can people find out more about this product?
So you can just look us up online on our website, DoseMeRx.com. I'm on LinkedIn. If you have additional questions, if there's anything I can answer, certainly love to connect with everybody. Where the world is getting more global and smaller. So I'd love to kind of talk to pharmacists in other parts of the world and learn about how they're practicing pharmacy and how they're incorporating various technology in their clinical care. So yeah, you can easily find me on LinkedIn and would love to connect.
Thank you very much. It's also on our Emerge LinkedIn page. You can just connect with her through that. Thank you very much for joining us today. And thank you very much for the questions that came through. And hope everybody's knowledge has been filled, and we can refill your cup next week on Wednesday on the next webinar. Thank you very much, Sharmeen, for everything, and hope to work with you again very soon.
Thank you. Bye-bye.
Thank you, everybody. Enjoy your day. Bye-bye.